First, you must realize that you have no idea before you can know the idea. We scan across all the frequencies if we want to learn anything new. Let us begin.
What has physics done for me lately? Second more, the equation E is equal mc squared. We have now acquired a fateful power to alter and to destroy nature. That's like when you're in physics and you go to a dream world saying, this is a physics excursion. Science, what is it all about? The whole of human history all falls in the dust of one stroke of a nail file. You can't really get to grips with evolution unless you realise what an enormous amount of time our own planet is only a tiny part of the vast cosmic tapestry, a starry fabric of worlds yet untold. You're tuned in to what can only be described as the best radio station on this blue dot we call Earth. It is, of course, 4ZZZ, connecting and amplifying voices of our community, be it on your conventional wireless radio by tuning into the classic frequency of 102.1 FM, digital devices such as DAB or smart speaker, listening via the Community Radio Plus app, or streaming us live from our sensational website at 4ZZZ.org.au. And of course, you can always listen back to our show, or any 4ZZZ show for that matter, using the ingenious on-demand feature also found at that URL. We also now have a weekly podcast of the show for your listening pleasure, curated by Toby, a condensed version of the show without the music, which my mum prefers. Just search for our show name, which is, of course, No Idea, spelt with a K, your weekly dose of science. And joining me today to deliver some gold standard science are two of my favourite science communicators. May I please welcome back after their hiatus of 2 or 3 weeks. Good morning, Peta.
Good morning, Max.
And I'm not meant to say good morning because the podcast doesn't work well then. And hello, Gabe.
Well, good morning to you, Max. You sound as tired as my voice sounds.
You sound very interesting.
After 2 weeks of paneling on your own, it's taken it out of you, Max. We're back.
Can I just run through some of the topics we went through from
Please
the archive?
Go ahead. Fill us in.
What did we go through?
Okay, Peter, you spoke about fish parenting.
Oh, good. Yeah, no, it's an important subject, and we all know the juvenile fish must be taught the ways of their elders.
Jay spoke about social height, not just diet, apparently.
Oh, I remember that one.
Yeah. And then, Gabrielle, you spoke about growing moss on Mars and why, why? Because it's not edible or anything, but you thought it might be a good idea.
Well, moss is pretty. Might greenify the red planet.
Some marine science from you, Peter. You spoke about shark teeth because they're the only fossils left from sharks because they're all cartilage.
Yes, because they don't have bones.
Yeah.
Yeah,
and then
pretty normal.
Gabe spoke about exam stress, especially in unfamiliar surroundings.
I remember that.
Issy spoke...
And they had the taller roofs.
Yes. Yeah.
Changed people's
ceilings.
Results.
Yeah, totally. And we spoke about how those rooms are always so cold as well.
Yes.
Unwelcoming,
freezing.
if you will. Issy spoke about some space food and the lack of taste on the ISS. Peter, bottlenose dolphins and their whiskers that disappear, and suddenly they can use those for electrical signalling, and they can find fish underneath sand in the studies.
Oh, yes. I was just imagining a dolphin with a beard and got distracted for a second.
I spoke about this microphone built on spider silk and sheep fly strike Gabe and the Australian goannas. Do you remember that?
Yeah.
Yeah.
Now, Max, today on the show,
go for it.
I've got some 9-legged male tarantulas for you.
Okay.
Oh. Oh.
Yep. What do you got, Peter?
I've got some rubber hands and I've got some fish treadmills.
Really?
Nice.
What have I got? I'm just doubling down on your story about the cheap date with apes, with the fermenting fruit on the forest floor. Do you remember that study?
Yeah, I do.
Yeah, they've given it a name now. So. Master Peter, what do you got for us?
All right, Max, question for you. Do you know the rubber hand trick?
Is that like the pencil trick, wobbling the pencil in your fingers?
No, okay.
That's pretty good.
I can't do that, though. Oh, wait, no, that one I can. No, I'm thinking of spinning a pencil. I can't do that.
Is it similar to that? No.
So not similar to that,
Isn't that
actually.
cool?
Yeah.
How does that work?
frame rate of your eyes. Okay, on to the next question. I first saw the rubber hand trick on QI, my favourite show of my youth. I've watched every single episode. And essentially, they put out this little contraption, right? Gabe's now wobbling a pencil. This is not important, Gabriel. So they put out this little contraption, and it looks like an upside-down T made out of wooden boards, right? So board on the table and then a little wall. You put it on the table in front of you, and you also put both of your hands on the table in front of you. Now, on the side, you put the board, this little contraption, let's say on your left side.
Yes.
And on the side of the contraption closest to you, there's nothing. But over the wall, there's a rubber hand. And it looks pretty fake. Like, it's usually a different skin tone. It's got painted nails. I don't know. It doesn't always look like your hand. It just looks like a rubber hand. Then there's also a big sheet that goes from the unit over your arm. So essentially, what you'd see if you were looking at a person doing this would be their real hand, then their real hand on a board, a little wooden wall, and a rubber hand.
And the real hand's covered.
Well, no, the hand isn't covered. The arm is. Your arm is covered.
Yeah.
You can see your own hand, and then you can see the rubber hand as well.
There's a couple of different versions, isn't there?
A couple of different versions, but most of the times you can see both of your hands and a rubber hand, you know? But yeah, and then a big sheet that goes over your arm, so you can't see your arm meeting your hand.
Yeah.
So you pop your hand in the blank space, and then some other person starts stroking both your hand and the rubber hand with a brush at the same time, in the same sort of methodology. Just brush, 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 brush. And after a remarkably short time, you get this.
Faster. I think that will help.
Okay, I got it. Faster is better. Keep it steady.
Right.
It's my hand.
It really does feel like
It's my hand now.
Yeah, after a short time, it'll feel like your hand. Your brain decides that is a part of your body.
Yeah.
You can feel the brush on your hand. And then all of that, I will say, I don't think I explained that quite as well as Stephen Fry, but it's quite a high bar to jump to. But all of a sudden, as soon as it's your hand, bam, they hit the fake hand with a mallet. And that is the test.
Really?
Not whether you feel the hand, but, in humans, we can just tell people that we do feel the hand. But the test is whether you jump. Whether you jump when the fake hand gets hit by a mallet. Are you so convinced it's your hand that you will jump and react to incoming pain?
What I feel like this is about to become about the ocean somehow.
What a great question, because I'm not your friendly neighbourhood psych, though I did walk that path for a small amount of time.
Yeah.
I am your friendly neighbourhood marine scientist. So which marine creature are we talking about today?
Octopus.
It's right, an octopus. Yes, Max. We're talking about an octopus, my favourite marine creature. An octopus fell for the hand illusion, except obviously it was a tentacle instead of a hand. Although I would love to see if they fell for a hand illusion. Anyway, now this amazing, wonderful, brilliant study is brought to us from the University of Ryukyu in Japan. QS ranking, please.
Oh,
302.
220.
401 to 450.
Yeah, I win.
Although I don't know if that's 2026 rankings. I couldn't find it on the 2026 website. And this was published in Current Biology, so it's very real. I want to point that out. This is a real, serious study.
Yep.
And essentially, they wanted to do this because they were interested in whether non-mammals had experienced this sense of what they call body ownership. That's what this test is testing, body ownership. Do you feel like that hand is your hand? Do you own it? And they wanted to test if octopuses could also be tricked into perceiving that fake arm as a part of their body. It's important, critically, because octopuses' cognition evolved independently from vertebrates. So we know it can happen in mammals.
Right, okay.
But octopuses independently evolved.
Can two relatively intelligent beings
with
that
very different brains,
different backgrounds, do they both have this weird little fault? I guess not really a fault unless you're doing some weird test with a rubber hand, but have this same weird little feedback loop where you seeing the hand. Oh, thanks for subscribing.
Hell yeah! This is the perfect time to subscribe.
That was Simon from Wollongabba, by the way. Thanks, Simon.
Thank you, Simon from Wollongabba.
Yeah, does that carry across into different
it's like a feedback loop of essentially you see something that looks like you feeling something, so your brain goes, that must be me, I'm feeling it.
Yeah.
So they placed these octopuses called plain body octopuses. Womp womp, sad name for an octopus. Feel bad for those guys. And they put them in a tank filled with polymer beads, which, yes, is that thing that toddlers eat out of vases that they're not meant to eat. You know, the little clear things you fill them with water. Yeah, those things. They had them in the bottom of the tank.
Yeah, okay.
And then an octopus on top of it.
With water, yeah.
Yeah, so not their natural environment, but a clean one.
Right.
And then the octopus is just sitting there, looks like a small octopus, and they covered one of the octopus's arms with this black plastic thing, and on top of that, is a fake rubber arm. So basically the octopus couldn't see the real arm, but they could see the fake rubber arm. And I want to point out here, the rubber arm in this experiment does not look like, it's not convincing. It's completely white. It's like white silicone tentacle, and the octopus at this point is reddish brown. So it's not even like in the human ones, they sort of make the hand a skin color, not necessarily your skin color, but a skin color, and has nails and stuff. No, they did not try in this. They were like, here is a white rubber
it's a white
arm.
stick.
It's a white stick, basically. I mean, it looks like a tentacle. It's like a silicone tentacle, but still. And then they did the same thing. They simultaneously stroked their real arm and a fake arm.
What do? They hold the arm of the octopus down and brush
this
it with
I didn't know, because they're
brush?
very movie and they don't love to be touched, so they must...
It must have been held down.
I mean, they were in very small tanks, so I think they just kind of could get them wherever they moved to.
Okay.
But they stroked them. And then in the test conditions, they did 3 different things. They stroked only the fake arm, they stroked both arms out of sync, and then they placed the real and fake arms in different positions. Then, after a period of time, they pinched the fake arm using tweezers and recorded the octopus's reaction.
Okay.
Right? And it worked. It worked. The octopuses reacted to the pinching of the fake arm by changing colour, which is a common thing they do when they're in danger, withdrawing their limbs, so going like, or fleeing, like trying to get to another part of the tank, suggesting that the fake arm was integrated into their body schema, which basically means like how your brain perceives your body. But the one thing that was clear is that it was necessary to stroke the arms in sync. When they didn't stroke the fake and the real arm in sync, they didn't react. So they clearly, it didn't get pulled into their body schema. They needed to have a similar sensation on a real limb to be able to integrate it into their body. So that's clearly important to this. I don't exactly know how, but it is a feedback loop that your brain goes, I feel something, I see something, that must also be part of me.
Right. So your brain needs the octopus brain needs 2 points of truth. It needs to be able to see it, and potentially just being able to see like an arm near you getting hurt. There's a reason why you want to retract your arm before the signal gets to your brain. In certain
exactly.
circumstances.
That's pretty much it. Is you can't wait for the signal to get to your brain. So you need to be able to see it with your eyes and go, danger.
But that stroking need in humans and octopuses is showing that it actually needs to be registered as your own. Your brain needs to subconsciously.
If someone hit a mallet on a person a meter away from you,
I'd still recoil.
I would recoil, but it's not the same thing. Whereas this is, you trick your body into thinking that's mine, and then you hit it.
Something has to happen beforehand that your brain is subconsciously patching that into what it's recognizing as its own body. And then when it gets hurt.
Now, can I say what I thought was really interesting about this study? I'll go on to what they say is their interesting parts. But for me, one of the things we know about octopuses is people say they have 2 brains, right? And I want to be clear, when we say 2 brains, it's not like one brain like ours and then another brain like ours sitting right underneath it. They have a lot of their neural tissue in their brain, but the vast majority of their neurons is in their body, in their arms specifically. In all of their arms, they have a lot of neural tissue that forms essentially a second brain. And we've talked about this before. It helps them taste and feel and react independently of the brain in their head, for lack of a better word. So they don't actually need to send things back to the brain. So that's where I find this really interesting, because the reason humans do it is because we only have one. We have a central brain. So it's useful for us to be able to make this body schema so that we can react without having to send a signal all the way up into our heads. But octopuses shouldn't need to do that anyway. So I wonder why they have this. Like, what is the purpose of this if they have that integrated brain in their limbs? Do you know what I mean? Okay, but then on to what the researchers say. The researchers are very excited because they seem to experience body ownership through multisensory integration like we do, and it suggests that they form internal representations of their own limbs. So essentially, like what I was talking about, it's called a body schema. It just means the map of you that your brain has. Your brain has a map of your body. They seem to have a map of their body in their brain too. And such self-awareness is clearly not exclusive to animals with backbones or similar nervous systems. Because like I said, very, very different brain style, really different shape, really different way of working. So it's clearly really far back, which again offers strong support for the idea that complex cognition evolved independently in cephalopods. Because we know like their close cousins, mussels, oysters, things like that, do not have this, clearly. Clearly, it's an oyster.
If you put a dead oyster shell next to another oyster and hit it with a hammer, it doesn't care?
No, I don't. I mean, it's not been tested, but I would really doubt it. They're not super highly advanced creatures, but we know that cephalopods are, so octopuses, cuttlefish, things like that, and that's evolved afterwards. So this is us both evolving this trait separately. Very, very interesting. And so, yeah, they want to keep looking at this. They want to they want to try other illusions, which essentially is just tricking more octopuses.
Yeah.
I have to get in this lab. I have to. I just want to trick octopuses all day long. I also wonder whether the octopuses go to the other guys and they're like, dude, the craziest thing happened to me today. You would not believe I got tricked. I thought this white tentacle was mine. Like, so strange. But yeah, I think this lab does some really interesting stuff, and I'd love to see what they come up with next. How else are we going to trick an octopus?
Brilliant. So back in the 7th of May, No Idea did a show. And you may
Chimps
have heard.
are known to share some foods, but not all
like
foods.
more
Not a
resource
lot of
guardy.
foods. They're a bit resource guardy, and when there is sharing, it's linked a lot to social hierarchies and intimidation. There's a lot behind it.
You gotta
Whereas in
earn
this,
it. Yeah, okay.
yeah. And there's sort of an order to feeding on
Yeah.
the best stuff. In
Fish do that.
Yeah, and in this they didn't see any of that. They just saw
'Cause they're
they
drunk.
just saw them feeding with potentially, Matt. So they just saw them feeding without any sort of forced behaviours behind it, right? They were just seemed to be happy sharing. When they went to test the fruit themselves, they found that the highest alcohol rate was 0.6% in the fruits. So it's hit the ground, and like you said, Max, the fruit's been there for so long that it has started to ferment and become alcoholic.
But it's like kombucha level. It's not really alcohol.
Right, so 0.6%, that's the highest. Most of them were lower than that, 0.25-ish, I think was
It's
the
just a bit
average
fizzy.
of some of the ones they were really liking. And like a sixth of a standard beer? Standard beer is about 3.5%, so this is 0.6%.
So it's a cheap
Lightweights.
date, really.
It's a very cheap, date.
You're thinking of dating apes? your next endeavour, Max?
Stumbling across a tree which occurs very commonly in their habitat and splitting a rotting fruit on the ground.
Wow. What a
Romantic.
Yeah.
So as we know, this feasting activity has a new name. Gabe.
It does.
Yes. It's called scrumping.
Do you like
Sorry.
it?
Why?
Scrumping.
That is,
Did they give any explanation?
Yes, they
Or
did.
did they
There's
just
some.
make a
Yeah,
word?
there is. Yeah.
It comes from the German word schrimpen, which means shriveled or shrunken when describing fruit.
And to scrump is
Do
also.
Germans use that? They go, oh, look at this shrimpen apple.
Yeah.
Yep.
Wow, okay.
And if you look up the word scrump now, it says to take fruit away.
Take fruit away.
Yeah, it's to gather or steal or share fruit.
I feel like you do that frequently, Gabe.
fermenting on the ground.
You're a huge fruit bat.
I'm a scrumper
You're a scrumper for life.
at my core.
And this research comes out of the University of St Andrews in Scotland. QS ranking, Peter.
113.
Oh, I was going to guess.
Also Dartmouth, though.
Yeah.
Dartmouth.
Yeah.
82.
I don't think it has one, does it?
247.
Does it?
Never
Okay.
mind.
Yeah.
Where's Dartmouth?
US.
Yeah.
Oh, sorry. I was thinking in the UK.
Dartmouth.
Yeah, I
College.
should have put it
You
in
want to
US.
walk us through this, Gabe, or
I can do a little bit.
Yeah.
So we saw that. The thing that I remember saying at the end of that study, where there were these chimps in Western Africa were eating alcoholic breadfruit and sharing it, was that they then started making these big, beautiful leaps at the end about how, well, we have common ancestors a little under 10 million years ago with chimps. Maybe those common ancestors evolved some way of consuming alcohol, ethanol particularly, in fermenting fruit, and then that was actually what led to the feasting that we see in humans and in chimps, and also potentially to the social drinking culture that we see in so many human
Alcohol
cultures
made us a
around the world.
society.
Right, rotting fruit, alcohol and rotting fruit may have made us a society, and so
Where else do you
it's a huge leap. But yeah, this new study takes us a little shuffle down that path towards establishing some more evidence behind that.
yeah, because they examined the dietary habits of orangutans, chimpanzees, mountain and western gorillas in the
Yeah.
wild.
Which ones will be closest to again?
So humans,
Chimpanzees, right?
So chimpanzees and bonobos are the most closely related on our branch of the great apes, and then we have a common ancestor with chimpanzees and bonobos. So the three of us have a common ancestor. I
Gotcha.
think on a genetic basis, we actually have a bit more bonobo than chimp, but pretty similar. And
Well,
then
that's upsetting.
The
I would
gorillas,
prefer to be a
the
chimp.
different gorillas are obviously very closely related, and then we all go. So we share a common ancestor with the chimps, the bonobos, and the gorillas a little under 10 million years ago.
Yeah.
And there's-
That is not that long.
Yeah.
no, and then about 10 million years ago is when they're thinking that one single mutation to a single amino acid may have come in one of our common ancestors that allowed us to process alcohol much more efficiently. Exactly. But yeah, Max, the dietary stuff they did in the apes, there were thousands of hours of footage and thousands of reports they were looking at for this stuff. It was huge.
Oh my God.
And they found that the orangutans can't digest alcohol nearly as well as the chimpanzees or the...
Does that mean they get drunker or they just can't
They just
can't
can't
drink
do it.
at all?
They just don't do it.
They don't do it. So
They avoid it.
0.8% of the records of orangutans had scrumping. The rest was not. So scrumping, foraging for this fermenting food on the ground.
Right, but I mean, like, if they accidentally ate rotten food, would it affect them more?
They don't share that same common ancestor. So then if you go back to orangutans, our last common ancestor, which includes the common ancestors with gorillas and chimpanzees and bonobos, with orangutans, is like a little over 15 million years ago.
Wow.
Yeah. But yeah, Max said the mountain gorillas, I think 25% of the time they were scrumping.
Yes.
The western gorillas, 62% of the time. The chimpanzees.
62% of the time they're eating their
fruit off the ground.
Yeah.
And
Damn.
31% of the time chimps. And they were saying it was they had to go back and do this work because no one's been looking for this before. This is why they gave it a name.
Right.
No one's been looking for it. We have this renewed interest, like
Yeah.
that study with the breadfruit in the Western African chimps.
Yeah.
But no one writes down fruit was consumed off the ground. They just write, feeding event seen or eating this type of fruit in this area. No one writes, was it on the ground? Was it in the tree? Anything like that. So
Was
to
it
go back
wrinkled?
through- yeah,
That's
so
right.
they had to go through all these records.
Wow.
So that's a good way to do it, isn't it? And then they say that the chimpanzees consume about 4.5 kilograms of fruit each day. So they suggest...
That's your dream, Gabe.
They suggest
That's your dream.
they
Imagine.
they ingest a non-trivial amount of alcohol.
Okay, can I ask a question here, though? Because, yes, that is a non-trivial amount of... First of all, okay, two questions. First question, are most great apes then walking around slightly
Half
sauced
cut.
all the time? Slightly sauced. I'm not saying they're sauced, I'm saying they're slightly sauced.
That's what they want to do next.
Yes,
is go
exactly.
And,
Test their blood alcohol level.
no,
Just
it's
breathe into
test-
this tube for a second,
test
please.
the alcohol level in the fruits, which is what they did in that study
Okay.
a few months ago. They found.
I
what
do
was
think
it?
they should
0.25,
breathalyze apes.
usually percent up to 0.6. But if you're eating 4.5 kilos of
That's a
stuff
lot.
that's at 0.25% alcohol,
You
that's
got to be a little sauced. Maybe that's why they're so
aggressive.
aggressive. They're just sauced all the time. I've forgotten my second question
It's
now.
alright.
Oh, my second question was, do you think if we, like Jane Goodall's sitting around with her chimps and they're having a good time and then Jane Goodall cracks a beer and they're just looking at her like, wow,
Yeah, true.
she can handle so
Yeah.
much.
Beers have been given.
She was in the
to-
mist. by the way.
People gave apes beer?
I've seen something about this. I'll look into it and get back to you, but I'm pretty sure beers have been given to great apes in captivity to help with their gut bacteria.
Really?
I've
Okay.
got something in the back of my
I saw a
head.
really cool thing. I'm riffing now, but I saw a fantastic thing this week about a zoo somewhere, I think in Europe, where there was this one chimpanzee who had horrific morning sickness, and they only found out that it was truly morning sickness because one of the zoo vets was also pregnant at the time,
Right.
and gave this chimp her tea that she has for morning sickness, and the chimp suddenly started eating again after having the tea and like requested
Really?
the tea in the morning so that she
Yeah.
could eat. Isn't that crazy?
Bit of
Anyway, Max, what else you got for this?
ginseng. That's all I've
Ape
got.
alcohol.
That's all.
Yeah, it was. They're pretty excited about this, I think, because it shows that there's behavioral stuff to link the evolutionary bits. They knew
Yeah.
that single amino acid mutation happened about 10 million years ago in the
Yeah.
common ancestor of us, bonobos, chimps, gorillas, and a whole bunch of
And we
other
say
now we
thank
can
you.
have
species as
alcohol.
We
well.
say thank you.
And we say thank you to that ancestor 10 million years ago who tweaked that amino acid.
That one mutation has really made a whole culture that they have no idea about. If they could see bars today.
But the orangutans aren't in that branch and don't have that
Yeah.
mutation.
So they're sober.
And so
They're really cheap dates.
shown that all the great apes that they studied that have the mutation are regularly scrumping.
Isn't that good? So if you
And
have
the
the mutation
orangutans
to
aren't.
be able to sort of deal with alcohol and not get super poisoned, but only a little poisoned, you actively seek it out. You're like, I'd love to be just
Or
a
that
little
you can
poisoned.
after. Yeah, you can suddenly seek it out and use it. And they're saying it could there could be two big evolutionary drivers for why you'd want to rather than just go up into the tree and get the fruit.
Yeah.
One being you
Seems
don't have to
easier.
compete with monkeys that are up there, so the other primates.
Damn monkeys.
So the great apes don't have to compete with them for the fruit. And the second being in a tree is really dangerous for a large
for an
animal.
ape.
I'd assume so, yeah. I don't like being in trees that much.
And it's one of the driving reasons why we became bipedal and our ancestors developed so much of what now makes us different from so many other animals is getting out of the trees. So
Yeah.
fantastic stuff. Amazing. We're going to keep following up the alcohol in
Exactly.
great ape stories
Yeah.
because this is awesome.
Alcohol made humans what we are.
I think what we're learning generally at the moment is a brain really isn't necessary for thought or learning. Like, I think we knew for a long time
That
that you didn't
explains
need a
me,
brain
then.
to react. It does. Oh my goodness, finally.
I can hear you. I can hear you gearing up to that.
Yeah, because he doesn't have a brain, so he has to make all that noise.
His arms and legs had to come to a committee agreement to make the joke.
You tuned into 4ZZZ, and the show is No Idea with me, Max. We got Issy in the room.
Hello.
We got Peta. We got
You do?
Gabe. What do you got for us, Gabe?
Max, there's no easy way around this. I've got to say at the top of this one that we're talking about spider appendages, so if you're not keen on that, you might need to tune out for a couple of
The
minutes.
extra leg, nice.
The ninth leg on a couple of male tarantulas.
I didn't even know they had these.
Yeah.
So, University of Turku is where this came
Turku?
from. In
Turku?
Finland.
Oh, I love the
T-U-R-K-U.
fins.
QS
Oh,
680?
424.
228.
366, but the difference between you, Max and Issy, and I think you're there.
I'm really off today.
So these researchers were looking at a particular group of tarantulas in the Arabian Peninsula and the Horn of Africa. So that's the Arabian Peninsula is the big chunk of land that contains Saudi Arabia, Yemen, Oman, UAE, Qatar, Bahrain, Kuwait, and then the Horn of Africa is the northeastern corner of Africa on the other side of the Red Sea from the Arabian Peninsula. So
Yeah, the bit
Djibouti,
just over where there's going to be
Eritrea,
a sea.
Djibouti, Eritrea, Ethiopia, and Somalia. So same general part of the world, just either side of the Red Sea in that region. And they had a whole bunch of specimens of tarantulas sent to them, collected and sent to these Finnish researchers. And then they analysed them in the lab, taking all sorts of high-def photos that entomologists have to take, or arachnologists have to take to do IDs and figure out the differences between the different species. And then they also did DNA analysis. And then they also looked at other types of tarantulas from places like Madagascar as a comparison to the ones from Arabia and Northwest Africa. So from that, they described 4 new species across that
Damn.
region.
That's
4 new
good.
species. And they're all fossorial, which means they live underground in burrows at the base of shrubs or between rocks. And they were so different, these ones, that they're actually an entire new genus. So
Oh, damn. Put that on your resume.
not just 4 new species in existing genuses. So for us,
Nice.
Homo sapien is our genus species. They've created a new genus of tarantulas to fit these 4 new species into because they're so different. And the name of that genus is Satyrex. The second part is Rex from Latin for king, same as T-Rex, king of the dinosaur lizards or whatever they're called. Rex, they've got Rex in there, and Satyr,
Well,
what's Satyr
satyr are
from?
goat people from Greek mythology, but I don't know if it also just means goat in Latin, but I'm pretty sure it means goat people.
It's from Satyr directly. Do you want to have a guess at why they've used Satyr in the name?
Do they have hooves?
'Cause they're never
quite
hungry?
Fluffy?
Nope.
Satyr, do they
have horns?
Early depictions of satyrs in Greek mythology.
Oh,
and
because
other
they were
mythologies.
very virile.
Very well gifted.
Very
Yeah.
well gifted. They were often used as like, oh, this woman in my village got pregnant, but satyrs though, I can't turn them down.
Yep.
Sort of vibe.
They don't put that in the kids' books, do they?
I didn't know that though.
So they've been given the name Satyrex, as in king of the satyrs,
Yeah.
because
wow.
the males of these species are very well endowed.
Right.
To the point where it's upsetting.
Well, so they don't have. It's not a penis in the way that.
I was gonna say, aren't they,
mammals
like,
have.
egged? They do
They're called
eggs.
palps, which are specialized appendages used by male spiders to transfer sperm during mating. And so, the largest of these species they described is 14 centimeters long. So quite a large
That's
tarantula.
huge.
You know, half the 30 centimeter rule you had in your pencil case at school.
It's bigger than most of the
Wow.
huntsmans in our house.
And the male palp can reach 5 centimeters.
Really?
It's just
Whoa.
5 centimeters.
a third of that length, and 4 times longer than the front part of their body, and almost as long as its longest legs. So,
Yeah.
you know,
Suffering from success.
Yeah, no.
suffering from
The Norway ski
is that
jumping team needs to
necessary?
get onto this.
Yes, they
So-
do.
the length of the palp was actually one of the key bits of evidence in them being able to say these are definitely different species. Really different. Because usually tarantulas have palps that are 1.5 to 2 times the length of their carapace, that front part of their body. These ones are 4 times, like twice as long as usual tarantulas.
You know, I've never looked at a spider and looked for its penis.
No. But Peter, you asked why. They have a tentative suggestion, they call it, as to why, which is a lot of spider mating is really brutal. It often involves the males dispersing large, that's why they're smaller often than females, dispersing large distances really just for the purposes of finding female mating. And if the female doesn't like the male at any point in the process, before, during, or after, they'll eat the male.
Yes, Slay.
So they
Yes, that sounds fantastic.
Their idea is perhaps they've evolved really long palps
So they can run away?
to avoid being attacked.
Love at a distance. Yeah, that's right.
Love at a distance. That's right, Max. That's what I got for you.
Is that
My genetic material.
a very short story for something very long? Nice, thank you.
Yeah. Gabe has a lot of penis facts in his brain, just generally.
That's going to be another little
Another sting for the show.
thing for the show. There we go.
All right. Now, the good thing is we may not have Max, we may not have Gabe, but we do have penis facts.
Triple Z.
We want to appeal to your senses.
You tune into 4ZZZ with me, Max. We've got Issy, we've got Gabe, and we've got our friendly neighbourhood marine scientists. Where's the sting, Peter? What's going on?
In press.
In post.
In review.
In review. It's happening. It is happening. By the end of this year, we will have. Okay, everyone. I've got a question. What does Nemo live in?
Anemone.
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 Anemone. Correct. Now, does anyone know how to correctly pronounce that word?
I
Because this is
thought
a
I
thing.
just did.
Anemone.
Anemone.
Anemone. That's
Anemone.
correct. It's not anemone. It's
Oh, the
anemone.
other. Okay, yeah.
Yeah. Common mistake. I didn't know the proper pronunciation until I started studying marine science in university. So don't worry, I might get it wrong in this story too, but it's anemone. So Nemo is an anemone fish, meaning he lives in an anemone. And there are actually 28 different species of anemone fish. Did you know that?
Oh, I've tuned out. I'm just trying to hear if you get it wrong.
Oh, great. Fantastic. Fact checker alive.
I'm trying to look up how to spell anemone. I'll be honest.
Anemone.
Anemone. Yeah.
Anemone. So there's not just Nemos, there's a whole bunch of anemone fish. So clownfish or amphiophrionine species, good lord, are renowned for their symbiotic relationship with sea anemones, which was thought to drive what's known as their remarkable ecological diversification. Now, I want to be clear here. When we talk about their diversification, we're not saying they are super diverse. Like, there's not like one fish that's got 3 tails or something, you know? They all look pretty similar. They're small. Most of them are orange or brown. A lot of them have stripes. They all live in anemones. They're all very aggressive. Fun fact, if you go and see these guys in the wild, don't get close. You will get bitten and hard. They're mean little fish. Nemo's not nice. And so these little guys, they're not that different, but they are remarkable because they went through something that's called adaptive radiation. Now, adaptive radiation is when there is a rapid increase in the number of species with a common ancestor, which is usually characterized by some event that has great ecological or morphological diversity, right? So these guys are all a little different from each other, and they split off about the same time. Now, for a long time, scientists have thought that this split and their diversity was due to their symbiotic relationship with anemones, and essentially like with these new anemones, they learn to live differently, blah, 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 blah. They each take
Lots
a different
of different anemones
species.
means there's lots of different clownfish
Basically.
that take advantage of each of those different anemones and the ecosystems they're in and all that sort of stuff.
Yeah, something similar to that, right? That's long been the assumption. And I think to a large extent that is still a big driving factor. But the researchers at Okinawa Institute of Science and Technology, sorry, no QS ranking, they wanted to test whether some other traits, especially swimming ability and muscle physiology, could contribute to the clownfish diversity beyond simple host specialization. Can I just point out that I messed up the word clownfish, specifically fish, but I've been doing fine with an anemone.
Yeah.
Awesome. Okay, so how did they do this? They got 14 clownfish species from around Okinawa, which, by the way, if you don't know where Okinawa is, it's only technically a part of Japan. It's quite far south, and it's got a complicated history. But yes, it is now part of Japan. Okinawa is a small island. It's got lots of beautiful tropical reefs around it. Gorgeous place. So they went out into the beautiful reefs off of Okinawa, and they found some anemone fish. And fantastic day, by the way. Yet again, another lab that just sounds fantastic to work in. They got to watch these fish all day, and they wanted to see and record how far individual anemone fish ventured from their host anemone. But then, critically, this is where they did some new stuff. They conducted what they called lab assays. So they did a bunch of lab tests. The first one, and the most exciting one, is that they did swimming speed measurements, which essentially they put fish in a fish treadmill. It's like, the way we see it is it's like a little glass tank, and it's got a checkered board at the back so we can see how fast they're moving on a high-speed camera. But essentially they just push water through the tank really fast, so they have to swim against a current.
Sure.
Fish treadmill. And then they also did oxygen consumption tests to see like how much energy they were using to swim in these currents. Then they did, I'm going to screw this word up too, microtomography, which is essentially a mini CT scan. So they put them in like 3 little mini CT scans. And then based on those scans, they did hydrodynamic modeling, which essentially means how good are their bodies at moving through water, right? Then with all of that data, they reconstructed their phylogenetic tree, or essentially their like family tree. Where did they come from? To analyze their evolutionary patterns and sort of test about like, okay, when did multiple species evolve the same thing at the same time, but not from a common ancestor? So convergent evolution. What came from a common ancestor? When did these things happen? Blah, 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 blah. And what happened was they found some ecomorphotypes. So essentially species with similar physical characteristics and ecology that were independent of their anemone specialization. So they have differences, even though they use the same anemones, meaning the anemone was not the factor that caused them to change. So they had 3 different types. They had the adventurous types. These are large, muscular fish with efficient swimming and go very far, capable of exploring away from their host. And these guys commonly had more than one anemone that they would live in as well. So they'd go around and live in a bunch of different anemones. Then there were sedentary types, smaller, leaner, higher metabolic costs, tightly confined to their hosts. They did not explore much. They didn't go away from their host much. They didn't swim very fast. And then they also had like a third group, which was kind of a mix of both. They didn't really go too much into this. But essentially what it shows is that their swimming ability and exploratory behavior were not correlated with the number of anemone species a fish can inhabit, which challenged the classic specialist versus generalist model. It was just a bunch of different things.
Which the idea would be if this fish can swim further, you'd think that it would be able to deal with more types of anemones.
Yeah, because it's adventurous.
If it can't swim further, you think it'd be really good at one type of anemone.
But that's not necessarily true. Yeah.
Okay.
There's a mixture. So essentially the anemone specialization didn't cause the genetic changes. The genetic changes happened independently, and some of them will use it. So obviously if you've got an adventurous fish, you'll see it visiting more anemones, but that's not the reason it's doing it, if that makes sense. And so, yeah, these morphotypes appeared independently multiple times across the evolutionary tree, which is convergent evolution. Basically, they split off, and then from that split, one became sedentary, like didn't move much, and one moves a lot. So, yeah, it's just sort of an interesting thing about how host specialization, like these anemones aren't the only story about clownfish. We can see them as separate from their symbiotic partners. But yeah, it's also, they just wanted to look at it because they find these sort of splits. Adaptive radiation is something that they're actively looking into. They've already done it with Darwin's finches and African kitchlids, which is another type of fish, sort of trying to see how this happens and where it happens. In the future, they're going to be looking at how these types impact their social dynamics, of which they are complex, their diet, and their interspecific interactions, basically meaning how do they interact with other species. So yeah, sort of an interesting study. Interesting because we always thought that clownfish and anemones were so linked together in their evolution. and seems like they might have some differences, that they still work together.
All new explorers must answer a science question.
Okay.
You live in what kind of home?
In an anemone.
Okay, don't hurt yourself.
Okay, it's time for the best part of the show. Loosely defined as science, yeah, you already know. Everybody listens to 4ZZZ just to hear us talking about what Butters just did. Subscriptions just keep rolling like the tires on a car. But something tells me that our science careers won't go far. But unlike an engine, I won't keep you in suspension. We're all here to hear him talk, so let's give him attention. You're not ready for when he starts rapping. Gonna hand the mic to Max, and I'm not talking Van Stappen.
Spoiler alert. Unfortunately, there'll be no Australian national anthem played today. F1 happened on the weekend. It was in Hungary on a circuit described as the Monaco without the walls. It was a trad race weekend. Traditional.
Are we really calling them that now?
Trad race?
Yeah, is
like
that...
trad race.
I just feel like it's mildly problematic. Yeah. That's all right. Whatever.
Which means the teams have 3 hours of free practice to get their cars sorted for Sunday's feature race. Then on to qualifying. Qualifying consists of 3 stages: Q1, Q2, Q3. In Q1, all 20 drivers hit the track to try and set a time that will get them through to Q2. Only 15 drivers go through into Q2 based on their best times. Then only 10 drivers progress to Q3.
So we lose 5 each time.
Here we do. And it frees up the track too. So Q3 is the most dangerous sort of, or the most cumbersome part of qualifying. Yes. You want to get a benchmark time in early on in case someone bins it. Mm-hmm. And then the whole session's red flagged.
Well, that's more likely to happen in Q1 because of traffic.
That's what I meant, sorry.
And
Q1, that's what I meant. Yeah.
Q1,
Did I say Q3? Oh, yeah, I meant Q1, yeah.
Q3, you get a little bit more room to play with, but you do have less time. So it's a bit of a trade-off still.
So would it be another McLaren front row lockout? And clearly no one told Ferrari's Charles Leclerc because he set the fastest time in Q3 to take the pole. Aussie Oscar Piastri managed to get position 2 and teammate Lando Norris P3. McLaren later said that their drivers were a bit too conservative in Q3 because there was a wind change, and they just wanted to make sure that they stayed on the track. The feature race...
a mega lap from Charles Leclerc.
As well, yeah, true.
Where did he pull that from? Meanwhile, where's Hamilton? Like, out in Q1,
Similar
right?
car, yeah, similar machinery, not working.
Crazy.
So talk us through the feature race, which happened on the Sunday.
Well, everyone sort of got off the line pretty well. But unfortunately for Lando Norris, into turn 1, I guess Piastri blocked him a bit, and suddenly he found himself down in P5, fast-charging Alonso. I was so disappointed when he couldn't quite get it to stick on Russell out of turn 3, but still amazing from Fernando there. Everyone kept it quite neat through that opening complex, though, like the turn 1, 2, 3 sort of section. No contact. Valtteri Bottas not on the grid, of course, so no one went bowling.
Yes. And George is so mature now with his driving style in the Mercedes. So
George Russell,
he was a bit of a menace for a while, wasn't he?
But now he's sort of a bit more control, a bit more thinks about it a bit more, and actually managed to keep within podium distance of Charles. I was going to say Verstappen. We don't talk about Verstappen much these days.
No, I was going to say, it was weird seeing, little bit of spoilers, it was weird seeing Alonso above Verstappen at some point. Yeah, so, interesting.
Yeah, he was fighting on basically the edge of the points. He was loving that chicane, though, wasn't he, for all the overtakes?
Yeah, nice overtakes, yeah.
Yeah, I don't think we saw anyone else use that, but that's one of the key overtaking locations. But no, everyone else was doing it into turn 1, basically. I think that was the only on-track overtakes we saw.
Totally, yeah.
So it was interesting. There's this split of, do you go 1 stop or do you go 2 stop? The track was quite cold, meaning that it could, like, the 1 stop was potentially viable, like start on mediums, go to hards, try and eke them out for the whole race.
Because tyre degradation is less on a cold circuit, I guess.
Exactly. And Piastri did the traditional 2 stop, which is so medium, hard, along with Ferrari's Charles Leclerc did the same. And then eventually Leclerc's car said no.
It did.
It just sort of gave up, and Piastri sailed past.
There's a few ideas going around that they might have put his tyre pressures up a little bit more for his second stop.
And that's coming out of
Did you hear
Alex
that?
Brundle, actually, the
Yeah.
younger Brundle.
Yeah.
Which is a really interesting theory, because he's definitely managing something on the radio, and generally the drivers aren't meant to be too public about, if they are managing an issue, to if there is something flawed in the car.
Yes.
But he was really on the radio saying, I told you guys this is the wrong idea.
Yes, I
would
can
have
manage
driven
it.
around it.
Exactly. Yeah. Keep
But
going.
meanwhile, Lando's just out front on a used set of hards, just sailing away. Piastri tries to fight back, but fortunately for our Australian boy, the Brits won it this time.
They did. So no national anthem. And George Russell really wanted Oscar Piastri to take out the McLarens so he could sail through. George was like 20 seconds behind these two cars out front, which is a long distance in Formula One sort of timings. And there would have been repercussions back at McLaren's head office if that had happened.
100%.
But Oscar was good enough. He locked up his front right going to turn one, but managed to stay away from Lando, so they let Lando win. Oscar second, and George Russell a distant third.
Yeah, it's like the days of 2014 again, where it's like Mercedes are like, how much should we win this by?
Exactly, and I wonder if McLaren's a bit like that too. Well, they wouldn't have been, because Oscar would have been pushing.
No, they were pushing. Pretty hard.
Yeah,
you can
totally.
see
And that's
that.
why they had the 20 seconds. Yeah,
just
True.
out of nowhere.
Okay, motorcycles happened on the weekend. Suzuka 8 hour. And it was a showdown between Japanese motorcycle manufacturers Honda and Yamaha. Honda came to the race as defending champions. Yamaha was hoping for an upset by bringing in MotoGP superstar Aussie Jack Miller as one of the three Yamaha riders. But it was not to be. The Honda team featured their own MotoGP rider, French rider Johann Zarco, and they were just too good and won the race. Yamaha was second. Not quite a Miller special, but Jack was happy enough with the result.
The Miller special is especially harsh, I think.
Gabe
Of all
coined
our
it.
running gags, I don't know.
NASCAR Cup happened on the weekend. Former supercar champion Shane Van Gisbergen was once again racing on an oval circuit, this time in Iowa, in full Red Bull livery. It was like he was driving his old supercar from the 888 days. Qualifying 21st and managing to get 8th on lap 169. SVG
Nice.
made a rare mistake, spinning and hitting the wall.
Oh.
Shane managed to get his car back to 16th position, but was subsequently tagged by Kyle Busch on lap 261. This ended any chance of him finishing in the top 20. Shane finished 31st. The Iowa race was won by William Byron, who managed to save enough fuel to get to the checkered flag, despite running on empty by the finish line, something that would never happen in Formula One. Why wouldn't that happen in Formula One, Maxi?
Well, because the FIA mandates that at least 1 litre of fuel be available at the end of the race, so they can actually drain it from the car and test it in case you're sort of cheating with your fuel. But of course, the last time I think we saw anyone get disqualified for this was Sebastian Vettel.
Really?
Oh no, it was actually George Russell, but that's all right. But at the Hungarian 2021 Hungarian Grand Prix, it was Sebastian Vettel. So that was the thing I wanted to mention about the one-stop v2 stop. Is it very much reminded me of Spa last year, with Russell going on the one-stop and Hamilton being the fast chasing.
Yes.
Merc
Okay.
behind.
Teammate.
Yeah.
And who
Anyway,
won that?
yeah.
Right? George win it.
Well, he did, and then he got disqualified because he didn't have the fuel.
That's great. Okay, F1, it's a summer break now, so we'll see the teams and drivers take a well-earned break for 3 weeks. And another FIA regulation for the F1 sporting code. All competitors, so all F1 competitors, must observe a shutdown period of 14 consecutive days.
And this is basically the only holiday they get.
Yeah.
That's it.
And then Radiothon and 50th birthday, because that serves as well, doesn't it, for no idea. So we don't have to bang on too much about Formula One and MotoRap next week because it's Radiothon.
Whoop, whoop.
We can give our full and undivided attention to this. It starts this Friday, by the way. And there'll be a myriad of pre-birthday celebrations culminating in the 50th birthday, which happens on the 8th of December. And the reason I mentioned the 8th of December is because the weekend prior, so the 8th of December is a Monday, so on the 7th of December, we get to announce, or they get crowned, world champion F1 driver for the year. And that may happen earlier, but
it may well.
Worst case, it will be after Abu Dhabi Grand Prix held on the 7th of December.
So fingers crossed.
Yeah, and they're saying that the Constructors' Championship which McLaren's going to win this year, could be decided as soon as Baku, which is like 3 races away.
Oh, that's
Crazy.
Yeah,
crazy.
and there's like another 10 feature races.
I suppose when you've got that many stacked under your belt, especially 2 championship winning drivers, the real problem is going to be which driver will receive the award.
Correct,
but as Max and I were kind of texting back and forth, our predictions are getting closer and closer, I think.
I want to hear some more anthems on No Idea. That's for sure.
Yes.
Anyway, any more to add, Maximillian?
I think that's it.
Easy.
No, I think if you subscribed during Radiothon last year, if you're like myself, you'll receive a little email being like, hey, just a heads up, it's time's up, time to get subscribing. So check your inboxes every once in a while.
Radiothon is approaching fast. From the 8th till the 17th of August. Get us a birthday present by subscribing to the station and make sure the next 50 are just as fun as the last.
That's 50 years in your ears. There's heaps of prizes, warm in a glow, and the sweet satisfaction of supporting independent community media. Make sure to head to 4ZZZ.org.au to subscribe.
That's 4ZZZ.org.au.
Community radio, baby.
On this day, back in 1991, the World Wide Web software was made public.
Oh, nice.
The software
Still
included...
deciding whether that was a good decision or not.
It included a platform-independent line mode browser, some web server software, and a library for developers. You tuned to 4ZZZ, and the show is No Idea. With me, Max. We've got Issy, we've got Gabe, we've got Peta. I meant to switch those around. What do you got for us, Gabe?
What I got for you, Max, is a pretty cool study that came out not too long ago about what may have been the first pandemic in humans.
Yes.
So this is researchers in Denmark.
The plague.
Yeah. Well, even before then.
Yeah, really.
They have found evidence from what may have been the first human pandemic. This was published in Nature and comes to us from the University of Copenhagen QS ranking.
Copenhagen.
88.
32.
60.
Close, Max. 101.
Oh, there you go.
Copenhagen.
There's an issue with studying old pandemics in that people who have...
Viruses don't last this long.
Well, people who have died or suffered from a disease thousands of years ago, shockingly, are dead and very dead.
Don't have many symptoms anymore.
Don't have many symptoms.
Their only symptom is that they're still dead.
Can't report much. And no one was around really tallying this stuff up.
Yep.
So you can ask them if they're in a pandemic, but they might not give you an answer. These researchers instead had to use an ancient microbial DNA from human teeth and bones. So they're looking at DNA, or looking for DNA from ancient bacteria, viruses, and parasites that are present in human teeth and human bones. Yeah, pretty cool. So the human specimens...
No, I didn't say cool. I said
The human specimens went up to 37,000 years old. From around parts of modern-day Europe and Asia. So we're not looking at Africa, we're not looking at Australia, we're looking at Europe and Asia in particular. They looked at 1,313 ancient humans and found ancient DNA from a human pathogen in about 3/4 of them. And there were a lot of different bacteria, viruses and parasites, 214 different types in all. They included, Peter, the bacteria behind the plague. The data showed the oldest known genetic trace of the plague bacterium in a sample from 5,500 years ago.
Yeah, the plague.
Well, no, the first plague pandemic happened 1,500 years ago. So the first bacterium sample they found was actually 4,000 years before that, sitting in a ancient human. And the second plague pandemic happened about 700 years ago. So already they found plague bacterium 5,500 years ago, which is a few hundred years beyond what they found evidence for before. And it's estimated to have killed between those plagues, estimated to have killed
Both
between
bubonic plagues?
between 1/4 and 1/2 of Europe's population across that Middle Ages period.
Yeah.
1,500 to 700 years ago. They found they found that same bacterium 4,000 years earlier in humans.
Wow, poor things.
But they didn't just find the earliest plague bacterium evidence. They also found the oldest known disease, known zoonotic disease, which are illnesses we get from animals, which include stuff like COVID-19, right?
So a lot of them.
A lot of them. And that oldest zoonotic disease in humans was detected 6,500 years ago, meaning it might be evidence of the first ever human pandemic 6,500 years before now. And that's several hundred years older than previous data had showed. And remember, they were looking at human remains as far back as 37,000 years ago. So they found the first zoonotic disease just 6,500 years ago. In that
Weird. So they weren't they haven't found the evidence of it, or they weren't getting sick like that before then.
Possibly didn't find the evidence, although they did have quite a good sample set throughout that time. So does anyone want to have a crack at why the first zoonotic disease may have popped up in this historic record 6,500 years ago, and then really taken off in frequency
Maybe
at that
our
point too? It wasn't a
population grew.
one-off. It was it started a spike after then. They didn't see any zoonotic diseases, so no diseases that we got from animals in these ancient European and Asian humans before that time.
Maybe we started farming animals and like getting closer with animals, or had a bigger population, something like that. Like,
I was thinking travel as well, like using boats and things like that, where obviously like you'd be coming into contact with at least like vermin, cattle, things like that. Trade routes,
around,
and that's hence spreading. What do you think, Max?
The timeline that they say it coincides with the best is that one on domesticated animals through farming and animal husbandry.
Hell yeah.
Nice.
Before then, humans in these areas were existing in a more hunter-gatherer context.
Wild
so
animals,
not huge
yeah.
numbers of people in the same place.
Spending lots of time with animals.
Well, before then, we're not talking about the animal contact in particular. We're talking about what that then facilitated.
Right.
So before this point, before we start working domesticating animals and using them in farming and animal husbandry, there weren't huge numbers of people gathered in the same place.
Yes,
Agriculture
of course.
facilitated and domesticating animals facilitated bringing people into a big place. Even if there was a disease outbreak before then from an animal, could have come from an animal, there was only so far it could spread because there weren't that many humans in close contact with each other. But once you get the agricultural breakthrough 6,500-ish years ago, or about 6,000 years ago, in parts of Europe and Asia, then that's when you see this zoonotic disease outbreaks start happening and potentially leading to these first pandemics. And the researchers say, if we understand what happened in the past, it can help us prepare for the future, where many of the newly emerging infectious diseases are predicted to originate from animals. Mutations that were successful in the past are likely to reappear, and this knowledge is important for future vaccines as it allows us to test whether current vaccines provide sufficient coverage or whether new ones need to be developed due to mutations.
That makes sense. I was going to say, if their thing was like, oh, we looked to the past to figure out how we fix this, I was like, I don't really want to go back to hunter-gatherer, personally. I feel like we're really locked into this city model that we have. I don't know if we could just suddenly decide to not do that. But vaccines sound good, yeah.
It's a pretty amazing study. So about 6,500 years ago was the first zoonotic disease, and then they really take off about 6,000 years ago, and have stayed at quite a high level since then. And then they also have the anthropogenic, so the human-born, human-to-human only diseases.
I wonder if colds got a lot worse then too, because I feel like everyone I've spoken to in the last month has had a cold, right? I don't know, have you guys had the same thing? Everyone has had a cold.
Not us in
Colds wouldn't
Brisbane.
have existed if we didn't have cities and
You
stuff.
say that now, Max. Don't forget, it's Echo weekend next weekend, so the Echo flu will be making its way around. But I think it
really
is
just the example of all of this wrapped up into one, right? The domestication of animals, bringing a lot of
I was about to say, there's a lot of
diseases.
petting. That would be really interesting, Peter. Yeah, seeing the evolution of the cold or the flu as well.
Yeah, a specific disease, not just
a specific
all
disease.
diseases.
Yeah, but I imagine that would be a little bit more complex.
Yeah, that's a virus too. Viruses are kind of like you said they looked at viral DNA too, because viruses do leave DNA. But I imagine it would be less than bacterial DNA. But I don't know. I don't know how the exact pathway of bacterial and viral DNA getting into your teeth and your bones.
To celebrate 50 years of 4ZZZ, we're calling on all our Zedders to get involved. As we recognize all the great art and design that has been a part of the station, we are asking for you to create a Zed. With Dissident Design Studio in cahoots, we are calling for a Zed created in any medium, dashed off, or painstakingly constructed to be a part of our celebrations. Let's build a visual celebration together from the ground up. Submit a Zed and it could end up anywhere. Head to 4zzz.org.au to find out more and submit your Zeds.
You're tuned to 4ZZZ, and the show is No Idea, your weekly dose of science with me, Max, Issy, Peta, and Gabe. And we thought we'd take this opportunity because it's going to be Radiothon next week. And they're describing it as 50 years of people-powered radio. And this will then culminate to a date, the 8th of December, because we first started broadcasting on the 8th of December 1975. That's 50 years of science right there.
Yeah.
And it also has the Sagittarius star sign, if anyone's interested. And some of the key traits of a Sagittarian, which is this radio station,
God.
It's adventurous and optimistic. Yeah, well, that works. Freedom-loving, yes. Honest and direct, yes. Philosophical and open-minded, well, sometimes. Generous and fun-loving, yes. And its weaknesses can be impatient, yes. Tactless, well. And prone to over-promising, well. Okay.
Oh, goodness. Really selling it back.
I can't believe we've gotten onto astrology on our science show.
So if you want to keep the Sagittarian station alive, if you're under 18, it costs you $25. If you're a pet, it's $25. Concession is $40. If a full subscription is $80, passionate is $140, or super sub for $500. That's a yearly subscription to 4ZZZ, and you can go onto our website at 4ZZZ.org.au/support, and you go into the running for a lot of prizes next week.
Yeah, I was going to say.
Starting this Friday, sorry. Yeah.
No monthly prizes because we are going through Radiothon, our big subscriber drive. And whilst you're at 4ZZZ.org.au/support, check out our new website as well. Yes. We've had a little bit of a facelift and it is super energetic for our 50th birthday. So if you want to check out all of that and more, 4ZZZ.org.au is where we'll head to you. We've also got new merch as well. So if you want to be repping us as you attend all our fun little gigs that we've got coming up for our 50th, then head to there.
Or just be a passionate subscriber and you get an...
you get it? Yeah, with some bundles.
T-shirt.
Yes, you do.
That'd be cool. Gabe, anything to add?
I'm checking out the new website.
I know, the new website was so cool.
Yeah.
Checking out what we look like on it.
So yeah, coming Friday, assuming it's the same system as we usually go through, there'll be a huge batch of prizes that people have incredibly generously donated, and you can sort of pick one and go into the running to win that in a draw if you subscribe during Radiothon from this Friday. So this is the time to think about it. If you're a regular subscriber who does subscribe during the Radiothon period, this is the time to start thinking about it, because next week it's all going to go off and that's your chance to resubscribe. Keep supporting this station, which does genuinely completely rely on the supporters, the financial supporters of 4ZZZ, and go into the running to win a huge and always incredible and awe-inspiring batch of prizes that people put up for offer.
No idea. Space news. ISS, an air leak on the Russian side of the space station is still a thing. Although the Russian crew hoped that
one
they
of those
had
things you don't want to leak out.
Yeah, I was going to say, that seems pretty important.
Seems relatively important. I don't know. Maybe it's low down on priorities.
Yeah.
They tried to gaffer tape it up, but long term.
Duct tape, I assume. Duct tape fixes everything.
Gaffer tape, yeah, duct tape.
Long term observations revealed that the ISS is still leaking air. During a recent press conference, Roscosmos official told reporters that recent attempts to fix the leak have slowed down the rate of air leaving the space station, but that is not yet completely sealed. So, job
it does seem like that's what you're aiming for, you know? We're not looking for slow release of air. We are looking for no release
Close
of
enough is good enough, I reckon.
Sure. Yeah.
Perfection is the enemy of good.
NASA's Crew 11 has arrived on the ISS. The Crew 11 quartet will join 7 people who are already living aboard the orbiting space station.
Entertain them? Did they bring their violins?
Yes, right.
Didn't think their sound would be that great up there, but you never know.
Question without notice: how many humans are currently in space?
22.
Oh, no.
I think it's about 8.
10.
Yeah. It's 14. There's 11 on the ISS and 3 on the Chinese space station. SpaceX's Super Heavy booster recently completed a static burn of its 33 Raptor 2 engines.
Can I just briefly interject here and say, every time we do this segment, I love space news. I hate SpaceX's names for their equipment. Super Heavy? Obviously it's super heavy. It's a part of a rocket. Oh, sorry. Frustrating. Just every time you say them, I'm like, that is terrible. It's just objectively bad.
That could be a new sting for the marine science. It will be
talking about rockets? Yeah, very relevant.
It will be used for the upcoming Starship test number 10 And fingers crossed, the launch should happen sometime this month. And finally, I'm going to give you a number and a name of something. It's called K2-18b. What do you think that stands for?
Exoplanet.
It is an exoplanet.
Oh, nice.
It's a supersized Earth, which may be our best prospect of finding life beyond our solar system. While signs of life on K2-18b have yet to be conclusively identified by the James Webb Space Telescope, the powerful Space Telescope has discovered that the planet, you're going to love this, is so rich in liquid water that it could be an ocean or Hycean world.
That sounds amazing.
It
Are
is-
you visiting soon?
It is
a little
not.
scuba set?
It's 9 times the size of Earth, which means it holds a heck of a lot of water. So, Peter, I reckon you should get excited. The
That'd be
planet
amazing.
may be rich with alien marine life.
I mean, it'd probably just be like the equivalent of bacteria, but I'm still hyped. Don't get me wrong.
Cool. And any more to add, Gabe, for the space news this week?
No, that's all I've got.
There's still space out there.
You've been loving the memes of Gilmore Space.
I have been, Max.
So what
Space
happened
4X is
for
cool.
those? It hovered for a while because 2 of its 4 engines failed to give enough boost. So it was only running on two half engines, yes. So
yeah,
that's
was
why
that,
it
were
only
the engines
hovered.
half full or half empty?
Well, there you go. I'm a half
Yeah.
full sort of guy. So next launch should be a bit more successful.
Yeah, and hopefully it happens soon. It is, Australia's first launch, Queensland-based Gold Coast Space Agency, Gilmore Space Agency.
They're
We've
around
had
Coomera,
we've
aren't
had
they?
a bunch of them on the show before, previously, chatting about some of their much earlier developments. The fact that we've sort of seen them come,
Yeah.
to the point where they're trying to launch stuff into space is pretty incredible. Even though it was a spectacular explosion very quickly after liftoff.
Womp womp.
Last week, but incredible to see what they're doing. Hopefully they can keep going more.
What do the next 50 years of 4ZZZ sound like? We have no idea.
Yeah. You tune into 4ZZZ, and the show is No Idea, but not for much longer. Sign us out, Gabe.
That is all we have time for. This last 2 hours of radio, or 1 hour if you're listening to the pod, has been brought to you by the word Scrumping and the lovely subscribers of 4ZZZ. We'll be back in your ears same time next week. Between now and then, give a fake tentacle a squeeze, tape up your air leaks and have a scrump. Those stories, you can find them on the 4ZZZ.org.au website. Listen back to No Idea, or you can find the No Idea podcast if you'd prefer. Just search up No Idea 4ZZZ on whatever podcasting platform you use. And I think we're still the equal top-rated podcast on
We are.
Spotify and Apple. A clean 5 stars. So, you can't go wrong there. We'll catch you next week.
And we'll speak to you next week.
Get ready for next week.
See ya.
Cheers.
I'm a goddamn marvel of modern science.